So this is a collaborative project covering out between the Clínica Universidad de Navarra, CIMA, that is a research center in between the program of gene therapy and neuroscience program, and the neuropharmacology department of the University of Navarra, and also with the Achucarro Center for Basque Research in Neuroscience. So the identification of the responsible gene in Dravet syndrome makes possible to raise its treatment by gene therapy. So to do this, we plan to use the high-capacity adenovirus, or gutless virus, that has a cloning capacity of the 37 kilobytes. So it allows not only to carry the sequence of the gene that is affected, mainly affected in this disease, that is the SCN1A gene, but also long extension of promoters to really control their expression, or other genes that we can consider that are important. So we love these vectors because they are able to infect both astrocytes and neurons, and we think that this is really important in the Dravet syndrome, because once injected in the brain, they remain stable for even 1 year after in rats. So it means that only injection in a patient could solve the problem for all their life. So apart from that, if you realize once injected in the brain, they cannot escape from the brain. And this is also something that we think that it could be interesting. So apart from that, they are non-replicative virus, so we can really control their expression. And this for us make them really interesting. So to generate a virus, the first thing that you have to do is to generate a cassette of expression that encodes for the protein or the gene that interests you and the promoter. So this is the first problem that we have to face with, that it was the generation of a sequence of the gene that wasn't able to mutate, because we realized that this gene has an incredible chance to mutate in the lab. So we recurred to a codon optimization protocol, and we generate another version of the gene. So after the amplification at the lab, we decided to perform a restriction map, and this will allow us to know if the gene sequence was conserved or not. So this was the theoretical map, and this was the real one obtained. So as you can see, it was conserved, and this was the first time that a lab managed to manipulate this sequence. So then we transfect this plasmid into the HEC line cells, and we checked that this sequence was able to produce the channel. And we checked it by real-time PCR, Western blot, and immunofluorescence, and as you can see, it was able. So this was the second problem that we had to solve. It was this channel functional or not? So to face with this, we recurred to the patch clamp technology. So we transfected head lines, and as you can see, the non-transfected cells weren't able to depolarize after electrical input. But the transfected with the plasmid that we generate with the SCN1 sequence was able to depolarize, and this depolarization was blocked by the tetrodotoxin treatment that is blocking the sodium current. So it means that the channel was working, and it was because of the sodium input. So then we are investing many times in the promoter selection, because we have to generate several kinds of vectors. So we can discuss about this later, but as you can see, we are proposing to generate ubiquitous vectors able to express the SCN1 gene in all the cells of the body. We are focusing also in neuronal specific promoters, in the endogenous promoters that will allow the expression of the virus in all the regions and all the cells that normally express the sodium channel, other promoters for the parvalbumin, and other for the GABAergic neurons. So with that, we will have a battery to control the expression of the gene of interest in all the cells of the body. So to start with, we decided to start by the CIS1, that is the CAG promoters. So it's an ubiquitous promoter that is going to affect all the cells in which the virus arrives. So we constructed the vector, the gutless vector that I was speaking before, with the CAG promoter and the sequence, codon optimized sequence. And after the production, that it takes like 3 or 4 months of production, we checked that it was able to infect the neurons. So we used the mouse neuronal primary culture, and as you can see, the infection with the vector increased dramatically the expression of the channel. But after the injection in the brain, and here we injected in 3 different points in the prefrontal cortex, hippocampus, and cerebellum, there was a huge increase in the expression of the channel. So the high-capacity adenovirus containing the SCN1 gene are viable and are able to express the sodium channel, and the problem is the stability. And we think that the first version of the vector that we used with the CAG promoter was not stable enough because it was a prokaryotic promoter. So now we moved to an eukaryotic promoter, that is this one, and we saw that it was stable for 5 months in the animals. So we are checking now the therapeutical potential of these new vectors. And for that, we are first characterizing of animal model that we just received at the lab. So apart from characterizing the incidence of the thermal induced seizures, the electrophysiological recording, and the motor, cognitive, and physiological evaluation, we are also checking for other aspects that the clinician told us. So with these results, we expect to be able to evaluate the therapeutic effect of the vector and have a wide range of parameters to be measured. So thank you very much.
Thank you.